everybody to this month's AKCI and KCAMA lecture series. We're thrilled to have Al Copeland, I just learned his new first name right now, from Mayo Clinic in Florida to talk about a story around renal cancer genes and sort of the translation and application of those genes to a phase 1 clinical trial. So this is right up our alley, for lab translational researchers and clinical researchers. And so, Al, we really appreciate your time and expertise again. I'm going to turn it over to you. And then, again, people can interrupt with questions, or we'll leave a little time at the end for questions, but the floor is all yours.
Brian said questions are welcome. So this is a story that started over 15 years ago when I was at UTMB in Galveston, Texas, and collaborating with Chris Wood, who was a GU surgeon at MD Anderson. And so we basically did genomic analysis, and this was the early days of genomic profiling using Affymetrix, human genome U133 plus chips. And so we identified the top 200 genes that are highly overexpressed in clear cell renal cell carcinoma, and you see this heat map to the right. All red is hot or upregulated genes versus patient-matched normal kidney tissue, and the list of the 195 genes that were upregulated in every tumor that we examined, regardless of stage 1 through stage 4. And then the question we asked is do any of these genes play a role in regulating cell proliferation as well as invasion and metastasis? And again, early on, this Sigma Aldrich had started developing shRNA, and so we did shRNAs, 5 shRNAs against each of the 195 genes in 6 renal cell lines, and asked the question, when these genes are silenced, which of them played a role in cell proliferation, and what we found was 31. Stop that. We found 31 genes that actually promoted cell proliferation, and a subset of these genes actually promoted metastasis. And this is a paper we published way back in 2014. But one of those genes that we cherry-picked out of this group to further look at was steroid-CoA desaturase 1. And SCD1 is the rate-limiting enzyme regulating conversion of saturated to monounsaturated fatty acids. So this is fatty acid metabolism is a cancer hallmark and critical in oncogenic processes. And essentially, what we see is the saturated fatty acids through SCD1 are converted to these monounsaturated fatty acids. And essentially what you're seeing is a loss of 2 hydrogens in the delta 9, delta 10 carbon, leading to this monounsaturated fatty acid. And these are 16 and 18 carbon. So the 16 carbon is palmitate, and the 18 carbon is oleic acid. And they're converted into all the different subtypes of fatty acids, such as phospholipids, triglycerols, and cholesterol esters. And these are used as alternative energy sources to glucose. They're critical and get integrated into cell membrane, so they're important to cell membrane fluidity and cell signaling. They interact and regulate the tyrosine kinase receptor activity and a multitude of other signaling pathways.
So I have a very quick question about the screen and the initial discovery itself. It's all being done in standard nutrient-rich DMEM?
Yes.
Initially?
It was, yes.
Okay. Yeah. And despite that, the lipid metabolic enzyme is having a significant survival effect in these cells?
We have since modified that, Abhishek, and realized that there are monounsaturated fatty lipids in serum, and the rest of the data that I will show you was done in cell culture in 2.5% serum, because there are monounsaturated fatty acids that will essentially, the end products are there, and so it doesn't matter whether you block SCD1 activity. So excellent question.
Thank you.
Great. So anyway, again, this is just the detailed reaction with regards to desaturating the delta 9, delta 10 carbon to get this monounsaturated. And again, the 18 carbon is the oleic, 18 is oleic acid, and the 16 is palmitoleic. There's a mistake here. My apologies, but this is essentially the detailed enzymatic activity. Importantly, there are a total of 4 enzymes here that are coordinately upregulated. And so this all starts with glucose and the TCA cycle in the mitochondria. And so acetyl-CoA comes out and comes into the endoplasmic reticulum where these other enzymes are located. And so starting with acetyl-CoA as the substrate, acetyl-CoA carboxylase is converted to malonyl-CoA fatty acid synthase, then converts it to palmitate, and then you've got SCD1 that desaturates and gives you the monounsaturated fatty acids and the double bond. And so again, this is occurring in the endoplasmic reticulum. And so this is, again, de novo synthesis of these monounsaturated fatty acids. If you look to the left here, you can also get from your diet, and again, going back to Abhishek's question, is, we have a novel SCD1 inhibitor that we've developed, and so we're going to have to pay attention to people's diets, right? Because essentially, olive oil is high in oleic acid, and so you've got, if you give it back through the diet, you've got cell surface receptors like CD36 that can transport these fatty acids into the tumor cells and make up for overexpression or the blockade of SCD1 and endogenous de novo synthesis of these monounsaturated fatty acids. This slide is busy, but what I wanted to bring to your attention is that the transcription factors that regulate these 4 enzymes that you see at the bottom, ACLY, ACC1, FASN, and SCD1, are regulated predominantly by SREBP1C, which is an oncogenic transcription factor. It's regulated and activated by an AKT-mediated pathways, and sometimes CHREBP coordinates with regulating these target genes. But in cancer, it's predominantly this SREBP1C transcriptional factor that typically sits in the cytoplasm and gets activated by an AKT-mediated pathway through cleavage and translocates to the nucleus to upregulate these sets of genes to promote synthesis of these monounsaturated fatty acids. This is essentially a common signaling pathway that's upregulated across all cancers. So all aggressive cancers upregulate this pathway. Rapidly dividing cells need to make these monounsaturated fatty acids de novo, because again, they're making new cell membranes. The palmitoleic acid gets incorporated into cell membranes. So and they also need higher energy levels. So they're utilizing these monounsaturated fatty acids for multiple purposes to survive. Now, if you block SCD1, SCD1 inhibitors can alter Wnt beta continent signaling activity, AKT, mTOR, the STEAP pathway. It leads to enhanced ER stress in endoplasmic reticulum and leads to an apoptotic cell death. Ferroptosis can also be upregulated through the STK11 KEAP pathway. It can also alter the immune, makes immune cells more immunogenic by blocking SCD1 and activating dendritic and CD8 T cells. And so there are multiple anti-tumor activity by blocking SCD1 in tumor cells, as well as immune cells, to affect tumor growth as well as cell migration and sensitivity to drugs. When I moved from UTMB in Galveston, Texas to Mayo Clinic, I helped recruit Tom Caulfield, who was a young postdoc at the time who did in silico drug modeling. And there were a few SCD1 inhibitors out there, and we couldn't get our hands on them. So I said, what the hell? And put Tom to work on trying to develop novel SCD1 inhibitors. And he came up with a list of 40 compounds. And of those 40 compounds, 4 of those compounds were actually SCD1 inhibitors. And this is from our 2018 off-the-target paper. And you can see we call it SSI 1, 2, 3, and 4. And you can see in the left-hand panel, there's no effect of any of these compounds on normal primary cells. So the HRE 152 is a normal primary renal cell. You've got myometrial cells, ovarian cells, breast, thyroid cells. They have no effect on these cells. Normal cells don't care whether you block SCD1 activity. And you can see a laundry list of other different types of tumor, from anaplastic thyroid cancer, the THJ16T and 29T, to triple negative breast cancer, to non-small cell lung cancers, to metastatic melanoma, ovarian cancer, pancreatic, and bladder cancer. So these are just examples of tumors, tumor cell lines that were screened and shown to be responsive. And so again, blocking SCD1 has broad activity against aggressive tumors. So our 2 lead compounds was SSI4 and SSI2. And again, I'm doing an enzymatic activity using mouse liver microsomes and radioactive saturated fatty acids. We can determine an inhibitory concentration needed to block the conversion of saturated fatty acids to monounsaturated fatty acids. And you can see that SSI4 has an IC50 of 1.9 nanomolar. And SSI2 has an IC50 of 29 nanomolar. Looking at the half-life, so SSI4 became our lead compound, and its half-life is around 3 to 4 hours after a single injection into a mouse. We also did a screen against 468 kinases and found no cross-reactivity, small with one kinase at 100 nanomolar. So highly specific and with very high affinity and specificity for SCD1. So our first publication was in clear cell RCC, and as you can see, looking at stage 1, 2, 3, and 4, we've got strong staining activity against SCD1 in all tumors compared to patient-matched normal renal tissues directly below each of the stages. And also by Western, you can see strong SCD1 expression and in RCC tumors, but not in patient-matched tissues. And down in the bottom right-hand corner, you can see the expression of SCD1 in tumor cell lines like CACI1, CACI2, A498s, ACHNs, And we also developed cell lines and PDX models. And so these 265s and 366Ts are cell lines that we had developed from patient tumors. And this was our initial demonstration of specificity with regards to inhibition of cell proliferation. And I think we were using 100 nanomolar of this. I'm sorry, this is shRNA. And so these are two different shRNAs, 780, and also rescuing the growth inhibitory activity by using exogenous oleic acid, depicted by OA here, and it's linked to BSA to make it soluble. And so you can see the oleic acid has a small effect on enhancing cell proliferation compared to control in the CACI1 cells. And if you treat with shRNA, you knock out about 90% of cell proliferation, and you can rescue it with oleic acid. And this is CACI1 cells and A498s. And in figure B, you can see SCD1 expression, and the shRNAs completely knock out expression, and you can see essentially no effect on cleaved PARP with oleic acid, and you can induce cleavage with the shRNA, and then you can rescue that cleaved PARP with oleic acid in the presence of the shRNA in both the CACI1s and the A498s. And figure C is just a picture showing knockdown of SCD1 and dead cells in the right column, and the oleic acid in the bottom right hand corner, rescuing the phenotype of in the presence of the shRNA. We also did it with an SCD1 inhibitor, A939572, and show a nice dose response, inhibition up through 10 micromolar dose of the A939572. And again, the same thing with the drug, show inhibition and rescue with oleic acid. And again, you can induce PARP cleavage with A93 compound and rescue it with oleic acid in the presence of the compound. And pictures again in the bottom right hand corner. We also looked at combination therapy. We don't really envision targeting SCD1 as a single agent therapy. And so we envision going into clinical trials with combination therapy, hitting multiple pathways. And this is timsirolimus. And you can see the blue line up to 50 nanomolar concentration of the A93 SCD1 inhibitor, and then increasing amounts of 1 nanomolar in the green line, 10 nanomolar in the purple line, and the light blue line here with 100 nanomolar timsirolimus. And so you can see a very strong shift downward and to the left, indicative of synergy. And you can see the FACI plot to the right, all below 1, indicative of synergy in cell culture. We then went to an animal model and treated with A939572 or timsirolimus, or the combination of the two in purple line, and you can see enhanced anti-tumor activity when the two are combined. And just looking at molecular markers of proliferation with Ki67, you can see attenuation with combination therapy in the far right hand column. Not really any effect on angiogenesis when looking at CD31. There doesn't seem to be any difference between combination or single therapy compared to the control. You can see cleaved caspase 3 is enhanced at least more than twofold in combination therapy. And you can also see downregulation of phospho mTOR activity in the combination therapy as well, and the timsirolimus panel as well. We also looked at CHOP, which is one of many ER stress-induced proteins in the endoplasmic reticulum, and you can see that it is induced with SCD1 inhibitor and enhanced even more in combination with temsirolimus. And so, just to remind you that again, SCD1 inhibition enhances, blocks the production of the monounsaturated fatty acids, enhances saturated fatty acids. They're actually lipotoxic and induce ROS and can be part of the toxic effect as well. And again, these ER stress proteins that are upregulated include BIP, CHOP, GRIP94, calreticulin, which is important in immune responsiveness as well through an ATF transcriptional pathway. And you can also see through the ASK junk pathway inflammatory pathways that are upregulated as well. So there's a lot going on when you block these pathways. So about this time, we actually became part of a liver spore here at Mayo Clinic and swapped gears to liver cancer. And so everything else you'll see is in liver cancer. So my apologies. But you got to go where the money is, right, Brian? So
can I?
Yes, please.
Before the segue, can I please ask you this? So I was thinking about this conceptually, right? We initially started. Excuse me. We initially started conceptually from the point of view of lipid dysregulation in clear cells, and that became an evident place to look for dysregulation of these pathways. But clearly, as soon as you start even seeing the effect in CACI1 cells, VHL proficient, HIF-low cells, ACH and VHL proficient, not typically clear cell type morphology, and then you're seeing the same phenotype in even the VHL deficient cells as well. You steer away from HIF-dependent lipid droplet associated dysfunction and now alternative mechanism. So conceptually, I'm thinking, as you're saying, lipotoxicity probably one aspect. The big struggle, and this is more of a SOS call essentially. The oleic acid rescues virtually every upstream impact that you can think of, from droplet dysfunction to alterations in many of these subpools of lipids. So in your mind, what could be other ways by which we could go down to the key node, that is the vulnerability leading to the fitness defects?
Say the last sentence again to me.
So basically, how do we pin the Achilles heel essentially? How do we what might be our biochemical tools to like tease apart the individual nodes over here? Because I feel oleic acid, I think the rescues are great, by the way. I love those experiments and the results are fantastic. But I feel oleic acid is so far downstream and such a massive master regulator that everything seems to be altered when the cells are fed with oleic acid. And so is there a way by which we could tease apart that, hey, only the lipotoxicity is the critical dysfunction, or only the lipid droplet is the key dysfunction? Do you think of biochemical or pharmacological ways by which we could be asking these types of questions?
We do have some insights that I can't share right now,
Okay.
but we do think we know what a master regulator is, and I'd love it. I had the slides in and took them out because there's some potential patent IP stuff. But yes, great. Do you have any thoughts, Abhishek?
Yeah, so in my mind, I think so, especially with the lipotoxicity aspect and the oxidative stress aspect, so much of that oftentimes feeds into ferroptotic readouts, and like oftentimes you get synergy with those types of molecules. The lipid droplets, Celeste has this story on HIF regulating the PLIN2 gene, and also there's some evidence of the HILPDA gene, so the hypoxia inducible lipid droplets. So that could be one in my mind. I don't know if there's a pharmacological way to do it, but at least genetically we could be inactivating those and seeing how they might synergize with SCD. Um, but we are struggling with this a little bit because we are working on a transcription factor separately. It's called NF1A, and we are seeing massive dysfunction in lipid-related pathways. We just don't know how to tease apart what's the critical reason why the cells are dying. It's too blunt edge of a tool to go after.
Well, maybe we need to talk later.
Anirban, did you want to? Do you have a question?
I just wanted to ask whether SCD1 blockade can lead to ferroptosis, but Abhishek partly touched my question. So yeah.
Great.
Okay.
Thanks.
So you'll see similar things in the liver story too. So again, we screened a large number of these cell lines in hepatocellular carcinoma, and you can see only a subset of these cell lines respond. And again, they're with an IC50 between 1 and 10 nanomolar from the responding cell lines. And again, in the top right panel, you can see that you can rescue this again with oleic acid. An interesting phenomenon that we discovered is that only really responsive cell lines grew really well on soft agar. So again, this colony formation, this sort of aggressive stem cell-like phenotype grows on soft agar. You can inhibit that growth with SSI4, and the cell lines, for the most part, do not grow that are unresponsive to SSI4. Okay, so it sort of suggests that we may have very high mitochondrial oxidation, respiration, and glycolytic phenotype in these responding cells. And so just using the seahorse assay, that's what we found. You can see here the green dots are the cells that respond to SSI4. They have enhanced glycolysis and as well as mitochondrial activity. And so again, getting at this idea of an aggressive cellular phenotype. We looked at unsaturated fatty acids, a very simple assay, just looking at not specifically monounsaturated fatty acids, but you can see much higher levels in responsive cells, and resistant cells have much lower levels of these unsaturated fatty acids. And again, another assay that demonstrated enhanced mitochondrial respiration and enhanced glycolysis, as well as lactate secretion in responsive cell lines. And again, we looked specifically at the ratio of monounsaturated fatty acids, specifically oleic acid, 18.1, versus stearic acid, and you can see very much higher levels in responding cells. And if you treat with SSI4, you can block that activity down to levels of non-responding cells. The non-responding cells do respond, but they don't care. They're not growth inhibited. They have not been shifted to some pathway that now is dependent on these oleic acids. Coming back to Abhishek's critical question, because this is what we've been wrestling with for years, trying to really understand mechanistically what's driving this whole pathway, and we think we know the answer now. So this is collaborative work from friends in Hong Kong. They were looking at in PDX models of liver cancer and combining it with sorafenib, a TKI. And you can see single agent activity in panel A with a dose response of this is our compound SSI4, 10, 50, and 100 milligrams per kilogram body weight. It's given orally on a daily basis. And And in panel C, you can see when combined with sorafenib, you get enhanced anti-tumor activity. And you can see the size of the tumors in panel D. And you can see, of course, enhanced ER stress through upregulation of another ER stress protein, BIP. This is work that we've done in PDX models of liver cancer, PAX148, which and as well as LIV58, which is a more aggressive tumor than PAX148. And the question, we were asking, do we see combination synergy with tyrosine kinase inhibitors? And here we're using cabozantinib and lenvatinib. Both are FDA approved for metastatic liver cancer. And the take-home message is yes, you can get strong, prolonged, durable response in these tumors when you combine SSI4 with a TKI in these tumors. We also asked the question, taking the resistant tumors to single agent lenvatinib and cabozantinib, can we take these same tumors that are now drug resistant to either lenvatinib or cabozantinib, and can we resensitize them by blocking the SCD1 pathway and treating with SSI4? And the answer is yes. So here you've got drug resistant tumors, and here's the combination of either lenvatinib in orange with SSI4, or in blue cabozantinib and SSI4. And so you can attenuate tumor growth. And we don't understand this mechanism at all yet either, but we're working on it. And so a short summary is this SSI4 has now been named MTI-301. It's been licensed to Modulation Therapeutics Incorporation. So just a quick summary, SSI4 is high affinity with high specificity for SCD1. It's got excellent oral bioavailability. It's well tolerated. It has broad anti-tumor activity against many cancers. It has in vivo single agent anti-tumor activity in PDX models. So we make lots of PDX models. So we've got PDX models in renal cancer and just about every solid cancer anybody needs or wants them. You're welcome to them. And so we've got anti-tumor activity in cholangiocarcinomas, liver, renal, melanoma. We've also demonstrated an in vivo synergy with tyrosine kinase inhibitors, mTOR inhibitors, proteasome inhibitors, as well as immune checkpoint inhibitors in immune competent mouse models. And so one of the areas that we're working with in triple negative breast cancer, as well as liver cancer, is combining SSI4 with immune checkpoint inhibitors as a potential to move into phase 2 trials after if the phase 1 trial is successful. And we do have some predictive biomarkers now that I can't really share with regards to, we can predict what patients are going to be responsive to an SCD1 inhibitor, as well as following patients. This is Modulation Therapeutics is the company, a small biotech company started by a friend, Lori Hazelhurst, a number of years ago, that we've licensed the compound to. We have been successful with garnering NCI STTR grants and now an SBIR grant to run the phase 1 clinical trial. And this is the team at Mayo Clinic. Dr. Han Tan was one of the co-inventors and with regards to our SCD1 inhibitor. Keith Knutson is a collaborator working with us on the immunology, and Winston Tan is a GU oncologist who many of you may know, who is the PI on this phase 1 trial. And Pooja Advani is a breast oncologist who will lead recruiting breast cancer patients, and Hani Babrik is a GI oncologist who will recruit GI patients. So we're recruiting in 3 broad areas in the phase 1 clinical trial. And so it's going to be run here locally on the Jacksonville campus. And these are acknowledgments of the laboratory and some funding and lots of collaborators. So questions, thoughts?
Yep. No, appreciate it.
Always
Open it up to the group for further questions and comments. I guess I can kick off with though.
Yes, sir.
Prashenda, go ahead. Prashenda, go ahead. Yeah, I just need to unmute.
Yeah,
There you go.
Hello, Dr. Copeland. So nice to
Good.
see you here. So I have one question because I think you showed like the inhibitor actually upregulates the Wnt signaling pathway, if I am not wrong.
You are wrong.
is-
You are wrong. It does the
I'm wrong?
opposite. Well,
That's the opposite.
the
In
slide,
that
the slide may have showed it, but actually it blocks the Wnt signaling.
Oh, okay. Then it's fine. I'm sorry.
Sorry, but yeah, it does. It also, interestingly, the soluble Wnt ligands, the frizzles, the very last step is a palmitoylation step that activates these ligands. So this is a mechanism by which it actually can block Wnt signaling pathway. So
Thanks, Rainier.
Yeah, I got a question. Yeah, great presentation. Very interesting. My wondering is if there's a kind of corresponding upregulation on the transporters in these cells for the fatty acid transporters, and wondering if that's could be a targetable.
we
Okay.
we tried to knock them out in a number of cell lines and only found, we first screened the cell lines and only found, this was in the liver cell lines, and only found one of them that really had high CD36 expression. We didn't look at the other transporters, Rainier, so we kind of stopped right there. But that was kind of our idea, was to block both
Thanks.
ways, and that might help us in a clinical trial with people who don't adhere to their diet. So this is going to be the real challenge, is whether you're going to be able to
Yeah.
be
So I have
successful.
another question. Sorry.
Sure, Chris.
So, yeah. So the idea is like cancer cells are very efficient at metabolic plasticity, right? So they can change based on the environment and like metabolic stress and nutrient stress. So how do you think like long-term resistance to this drug? Like maybe cancer cells will get adapted to lower oleic acid or something, the lower inhibition of this compound.
Well, what I can't tell you is we know what state we've got to put these cells in, and we know how to do it now. And so it has everything to do with metabolism. And so if they get out of that metabolic state, they will become resistant. So you've got to keep them in a certain metabolic state, and there's a switch to switch it. So
I have a very basic question going back, I think, to your first slide with that Affymetrix chip. And I think you found, I don't know, maybe 20 genes that were upregulated. How do you pick which one to pursue? I mean, is it, gee, we have expertise in this one area, we're going to pursue it, or are there other criteria? Because I always see, as more of a clinical drug developer, I see these, and it's like, we pick this one thing way out here that's, kind of looks like everything else to me,
I have a postdoc position open, Brian. I can teach you experimentally how to answer this question. There's a usual needle in the haystack problem, right? You end up with like hundreds of genes, and then you don't know what to do.
Right.
And typically you have to set up some kind of a secondary screen in which you will, among them, pick up the first among equals, if you will. The thing is, not all genes that are upregulated or downregulated are important in the context
Sure.
of the tumor.
Yeah, for sure.
So there are lots of bystanders, and so the key thing really is to determine which ones of those are functionally actually driving the phenotype. And so I think based on what I saw on the slide, I think you have done the screen in an arrayed format, right? So you individually knocked down the genes, Al, I think. But typically these days we would take, let's say, the top 200 genes. We would make a CRISPR library targeting all 200 of them, and then we would see which ones of those would be the topmost hits in our screen to fish out, oh yeah, this gene among those 200 is the most critical driver of survival.
Yeah.
So that's usually how we would end up.
I think Brian is referring to the 31 genes out of the 200.
That one.
Oh, yeah.
So here we got 31 genes.
Yeah.
Why did we pick SCD1 versus any of the other 31? And it was a long conversation, but Han Tun, who I showed his photo, he's an oncologist here, and Han was the one who essentially said, go after SCD1. It plays a critical role in cell metabolism. Nobody's working on that, and it also plays a critical role in stem cell phenotype as well. It's important in stem cell phenotype. So, and
also druggability, right? I think that's a critical feature. So there are many genes over here that you will find that are not historically in a pharmacologically druggable category. And so MYC is a great example, right? We know for years MYC is critical, but how do we target it?
At the time we were not thinking about that. We were just thinking about it. Interesting,
Yeah.
But in line with your metabolism links, and so the one idea that, I mean, is probably something that I'm certain you folks have thought of, is the business of how high-fat diet or environment is impacting the dependency of SCD1, right? So even in the RCC space, this is a little bit of a controversial space, I feel, in some ways, because we had all those obesity paradox papers where we suggested that there was increased incidence in people with high BMI, but also the tumors tend to be more indolent in nature. If you look at like mouse models, that really does not hold up even in clear cell, right? Animals that are fed high fat diets, the tumors grow a lot more robustly in those mice. And so that I don't know how much that clinical feature is being modeled in our ongoing experimental setup. So thoughts about how you would, what are you doing in vivo? Do these mice get regular like chow? Are they on high fat chow? How do we tease these aspects apart?
Yeah, so we're doing all of those experiments right now, and like I said, we know that high fat diets with monounsaturated fat, and remember, it's got to be monounsaturated fatty acids. They will drive tumor progression and enhance tumor growth and cause resistance to an SCD1 inhibitor. We know that. And so like I've been saying, to Ranier's point, is diet in the patients is going to be critical. If we can't monitor it and we can't keep patients on a low fat monounsaturated fatty acid diet, we're done. So we're, we've collaborating with a guy named Walter Longo at the University of Southern California, who's done these fasting mimicking diets, and we've asked him to take that diet, and they're actually high in monounsaturated fatty acids. And so what they've done is they've made one for us that drops the monounsaturated fatty acids, and we've done some initial work with fasting right now, and looking at all of these different parameters with regards to enhancing sensitivity to SCD1 inhibition. as monotherapy as well as combination therapy.
A question from Brian Lewis in the chat. I don't know if you could see it, talking about, I think you mentioned this at the end, but maybe just to reiterate the phase 1 plans and what it means to the patient community. So I think you said the phase 1 has started across disease types. Was that GI, breast, and renal?
It has not started. We're maybe 1 to 2 weeks away. Hey, Brian.
Okay.
So say the question again. I was too busy.
Well, it just says, can you discuss phase 1 plans and what it means to the patient community?
So it's standard phase 1 3x3 dose escalation in a phase 1 trial, and our plans, at least with triple negative, is if we get to, again, it has to get through all the toxicity stuff, but if we do get through that, we're anticipating a phase 2 trial in triple negative breast cancer with immune checkpoint inhibitors, and then with liver cancer, TKIs, and immune checkpoint inhibitors. And so those are the thoughts for phase 2 trials.
And the drug is now, it's owned by a biotech, you said, or licensed to a biotech?
It's
and they're
licensed
funding it.
to Modulation Therapeutics. Yes, sir. Yep.
Yeah. And they're obviously funding the trial. Yeah.
NCI is funding the trial.
So.
They are not cash rich, so we stayed very involved.
Rainier, you have a question?
Yeah. I mean, my team's just kind of acting now, but I related to the metabolism. Do you think there's gonna be any interaction maybe with the statins?
Yes, we know there is. Great question, Rainier. Yes, there is synergy with statins.
So that could curve a little bit the diet part, maybe.
Potentially.
All right. Okay. Thanks.
Yep.
Thanks, Rainier. Anybody else? Questions, comments? I mean, I think these metabolic pathways are super interesting. It's sort of, to me, sort of an untapped area mechanistically.
Yeah.
But with a lot of challenges, as you've outlined, and Abhishek and others, there's just a lot of challenges to diet, considerations and picking the right enzyme, and I think seems to me we're a ways from solving those. But I think as a category, I think they're exciting.
Hopefully, we'll have a nice story to share with you maybe in the next 6 to 9 months. We'll see. They're put together.
Yeah, we'd love it.
Nima is here, right? So Nima is on the call, I think. Yeah. Nima, I remember you folks were doing those preliminary trials with the metabolic kitchens and like trying to ensure that patients would be taking a controlled supplement. How are those studies ongoing, or have you all stopped? Can you?
Yeah, it's a neoadjuvant prostatectomy study. It's very, very difficult to enroll to, actually. Very challenging in the neoadjuvant setting because men are, newly diagnosed, and oftentimes they go out of the country prior to surgery. So, I mean, good reasons for having challenges with enrollment, but it's a different setting as well. So, very, very difficult, though. That's a great question.
Great. Thanks, Timo. All right, well, we really appreciate your time. Unless there's any more questions or comments, appreciate your time and expertise.
I have an extra comment, Brian.
Yeah.
What's that?
I have an extra comment. I mean, by the way,
go ahead.
Put a plug. We have an imaging agent that is a phospholipid. So maybe there is a way to get a readout on some of this status in patients. So maybe that will be interesting.
Tell us about your reagent, Rainier.
It's a PET imaging agent. So it's a phospholipid, phosphocholine.
Okay.
And it essentially enters through lipid rafts into cancer cells. It's a fairly pan-cancer agent, which kind of checks out with what you described. This lipid metabolism is high across the board. So send you some references if you're interested.
I'm very interested. Please send me references. My email is copeland.john@mayo.edu.
Yeah, it should be in the invite, Rainier. Let me know if you need it.
That's right.
It should be in the Outlook invite. So yeah, connect later. That'd be great.
Yeah,
Awesome.
no, yeah. Because
Thank you.
we also, I also helped start a small PET CT imaging facility here. So we're into creating radio label nucleides as well. And so we've got a cyclotron facility here and radiochemist. And so we've developed,
Awesome.
we've actually radio labeled SSI4 and with carbon 11 and done some other novel radioligands for diagnostics and theranostics. So very interested, Rainier.
Awesome.
Great.
Thank
All right.
you.
Yeah, thank you.
thank
We
you,
appreciate
everybody.
your time. Thanks, everyone. And we'll see you all soon.